everyone. I am ARPIT KARAN from B.Sc Life Science second year, and today we all will be going to learn about fate maps. And I'm sure by the end of this video, you'll have very clear concepts about what fate maps are. So बिफोर स्टार्टिंग पहले उसके बारे में जान लेते हैं कि फेट मैप्स होते क्या हैं. मोस्ट ऑफ द टाइम्स स्टूडेंट्स कंफ्यूज इट विद द फेट ऑफ germ layers by simply mixing with the words, but that's not the case always. We'll be going to study it in the next few slides. So I'll tell you what's the difference in it. They are two totally different topics. As we all must know that we started our life from a single cell zygote, एंड जैसे जैसे हमारी डेवलपमेंट हुई सेल्स डिवाइड करते गए, जाइगोट से एम्ब्रियो बना, एम्ब्रियो से एक फीटस, एंड देन वी वर बोर्न. हैव यू एवर वंडर्ड दैट how our complex organs are formed and how our complex organ systems are formed from only a single cell? So we'll be going to learn about that only. Fate maps are basically the diagrammatical representation, and they tell us about what a cell is going to form at the later stages of development. Which organ will it form? So let's get started. So what are fate maps? As I have already told, these are the diagrammatical representation of the fate of the cells, that which cell is going to form which kind of organ in our body. As you all know that when our cells align to form the germ layers, we already know that this germ layer is going to form this kind of organ. As the cells align to form the mesoderm, we already know that this will be going to form our heart. But if we want to know prior to gastrulation that this cell is going to make heart or liver or kidney, then what measures should we use? There we use fate maps. So fate maps are the sequence of fate maps at consecutive stages of development. These show the progression of different cells, that how the cells divide, how the cells multiply, and how the cells align themselves in the later stages of development. Why do we study fate maps? What is the significance of studying fate maps? First of all, they are very useful in embryological experiments, and they help to trace the cell lineage, that a cell is giving rise to multiple cells, then a tissue, then an organ. So we'll know that this particular cell is going to form this organ in our body. So it helps to trace the cell lineage in our body during the early embryo development. So, and it also helps in understanding the mechanism of morphogenetic movements. As you all must know, morphogenetic movements are responsible for the formation of germ layers. So we can also understand the mechanism of morphogenetic movements through these fate maps. Moving further, so there are different methods of mapping a cell's development. But what should we know prior to that? That why are we doing fate mapping? Suppose we have a mass of cell here. We have different cells, and we don't know which cell is going to form which organ, and we want to know it. So first of all, we'll mark that cell. For that, we use particular kind of labels which we'll be going to study in different methods of mapping. So mapping is something in which a cell is labeled so that it can be studied at later stages of development. So there are different methods of mapping, and I have also mentioned that what are all the characteristics a good label should have. A good label should not spread to neighboring cells. If we are using a label in a particular cell and it spreads to the neighboring cells, then we won't be able to map the particular developmental stages in that cell, and it should be readily detectable. It should not fade away at later stages of development, as we'll be going to study in the later slides, and it should not interfere with the normal cell development. If our label is interfering with the normal cell development, then how will we get to know that this cell is developing naturally in a particular way? So these are all the characteristics a label should have for the easy purpose of mapping it. So we have two methods of mapping. First one is natural, second one is artificial. Artificial has a lot of sub methods in it, and we'll going to cover all of them in our slides. First one is natural markers. It was discovered by E.G. Conklin in 1905. So what does natural markers mean? That is something which is happening naturally. We haven't made it. So in an organism, some particular pigments are present at later stages of development, which help us to map the particular cell fate in it. So a particular example is can be seen in tunicates. As we have already studied in animal diversity, ascidians, the fertilized eggs of ascidians, tunicate sea squirt, we can see this kind of marker. In this, the natural color differences in the cytoplasm of early blastomeres. So, uh, prior to gastrulation, blastulation stage is there. So, in that, the blastomeres have particular kind of colors in them, and through which we can map the fate of the cells. So, I have mentioned here that the cells which contain clear cytoplasm are going to form the ectoderm. The dark gray yolky cells will form the endoderm. The light gray will form the notochord and the neural plate, and the yellow will form the mesoderm. I have also attached a diagram there of the stages of development in a ascidian larva. Here we can see an 8-cell stage in which a particular portion is colored yellow. So, as we see in later stages of development, it is leading to the formation of trunk in that particular organism. So, we know that this cell is going to form the mesoderm in that organism, and that mesoderm is particularly leading to the formation of the trunk in that organism, as we already know. So, this is the natural method of mapping. Moving forward, we come on to vital dyes. First artificial method. It was discovered by It was discovered by Walter Vogt in 1925. So what basically are vital dyes? We stain the cells with it. So basically we have a mass of cells and we have to label a particular cell in it. So we use a dye. Basically like how we used to highlight our notes, which is something important. So we use dyes in it. The best part of these dyes are that it stains the cell but it does not kill them. So I have mentioned a few examples of vital dyes here, which contains Nile blue sulfate, neutral red, Bismarck brown, and Janus green. And I have also mentioned a protocol here that how can we use these vital dyes. First of all, we make agar mixture of a dye by mixing it in and it on a cellophane. We'll press it on the chosen area of blastula for a short period of time so that that particular stain diffuses into our blastomeres. Then when the stain diffuses to the blastomere, then we can easily study the cell movements which are followed within the embryo, as you can see here. But one drawback here is that vital dyes fade away. As our development proceeds, the dye seems to fade away. As you can see that we have put on a dye here, then these particular cells are labeled, and then it dilution takes place and this dye fades away. So it is very difficult for us to monitor the movements at the later stages of development. Moving forward, we have carbon particles. It is very similar to vital dyes, just that we were letting the dyes to fuse into the cells. In carbon particles, we let the carbon particles stick on the cell surface so that we can monitor the movements and the cell looks apart from the cell mass, in which all the cells look similar. So in it, we have two examples, and the examples of two scientists also who worked for carbon particles. First one was N. Spratt, who made the fate map of chick. In that, carbon particles were stick on the cell surface and the movements were monitored to trace the cell fate. And then William W. Ballard modified this technique. As we already know that carbon particles were sticking on the surface. So what did he do? That he injected the carbon particles into the embryo, into the teleost embryo, which is a fish for your knowledge, and he traced the cell movements to make the fate map. So here I have also put the picture of a chick whose fate map was made by N. Spratt, and this is a teleost embryo in which carbon particles were injected by William to make its fate map. We have radioactive markers. So what are radioactive markers? These are also an artificial method of labeling. I have also mentioned a few examples like carbon 14, phosphorus 32, and H3, which is tritium, an isotope of hydrogen. In chick embryo cells, these all were used, and through these markers, by the technique of autoradiography, the cell movements were traced and the fate maps were made. So, I have also mentioned here that tritiated thymidine labels the nuclei. When it is incorporated into the DNA of the cells, it labels the nuclei, and a region of interest is cut from the host then, and it is put into the and it is replaced by a radioactive graft. Like we have prepared a radioactive graft and we have put it in a host embryo. This is a host embryo, and by the method of autoradiography, we can trace all the cell movements and we can make a fate map out of it. Moving forward, my most favorite type, fluorescent dyes. As you all must be hearing from your labs about fluorescent microscopes. So it uses fluorescent microscopes. The first fluorescent dye was synthesized by Adolf von Baer. So what basically are fluorescent dyes? The major thing about using fluorescent dyes are these are large in size and these are inert, so they cannot pass on to another cells. So that's one thing about being a good label. And they do not as they are inert, so they do not interfere in the normal functioning of a cell. So what do we do? As I've already told you about the characteristics of fluorescent dyes, these are micro injected into one or more cells. Whatever cells we want to study, we micro inject fluorescent dyes into them, and the descendants of that cells, we study those to trace their movement and make fate maps using a fluorescent microscope. So here I have mentioned a few examples of fluorescent dyes like fluorescent carbocyanin dyes, indocarbocyanin and oxocarbocyanin. These are long lasting, and as I have colored it in two different colors, so this one gives the red color and this one gives the green color. And these are lipophilic in nature. As you all know that our plasma membrane is bilipid in nature, so these can diffuse very easily into our cell membrane. The last topic for today is the genetic markers. As we have studied about all the dyes, the carbon particles, they fade away with time and they are not able to give us that efficient results as these give. We make changes in the particular genome of an organism to study the changes. It is a permanent way of making cells and following their fates. So what do we do? Its advantage is that it does not spread to neighboring cells, and if it is stably expressed. First of all, we'll genetically modify those cells to study, and if it is stably expressed and inherited by the following cells, by the descendant cell, then it is very advantageous for us to study. But it is also very low in efficiency because the introduction of gene is by chance, so it does not always occur, and it is very less efficient process to do. One of the famous examples of this is they used to create chimeric embryos. What basically are chimeric embryos? Chimeric embryos are the combination of two embryos from two different organisms which are almost similar in their developmental stages, but they are totally different species. They are done by xenoplastic transplant. Xenoplastic transplant means that we are taking something from another species and transplanting it into another species. So, embryonic grafts are transplanted in a xenoplastic way from the animal of interest, which has a totally different genetic constitution and but similar developmental pattern, as I've already told. So by this genetic markers method, we have created chick and quail chimeras. As you all have studied about the developmental stages of chick in your previous classes, so chick, as we already know, and quail is also a similar kind of a bird, which is of different species but has similar developmental stages as of chick. So we made chick and quail chimeras using the genetic markers. So how they are made? They are made They are made by grafting embryonic quail cells inside a chick embryo while the chick is still in the egg. So our chick is still in the egg, and we, through xenoplastic transplantation, we take quail cells and transplant it into the chick's embryo to make the fate map of it. So why do we do it? Because there is some difference between chick and quail eggs. The proteins which are used to identify quail cells, they are different, and they can be easily identified at the later stages of development. So we use quail eggs, and one of the major reasons for using quail embryo is also that it is having similar stages of development. So chick and quails are developed in a similar manner, as I have already told you, and the grafted quail cells become integrated into the chick embryo so that they can participate and construct various organs in the chick. The chick, when hatches, it will have quail cells, and which can be identified easily from its immune system. By seeing that where these cells are migrating in a particular chick, so we can make the structure maps, the fine structure maps out of it. For reference, I have also put one image here in which here, as you have already seen, this is a quail embryo, this is a chick embryo. It is a donor, it is a host. So we have taken a portion of quail embryo, and through xenoplastic transplantation, we have transplanted it into the chick's embryo here, which is the portion which is responsible for neural tube and neural crest formation. When uh, these cells are incorporated into the chick system, and when the chick is born, we can study at the later stages of development the movement of these cells. As you can already see that these are quite easily identifiable, so we can create fine structure maps out of it. So that was all. I hope it was clear to all of you. Thank you.